Hello to my all dear students. Welcome to a new video. As the revision is going on, I will discuss some important topics in each video. In today's video, I will discuss the mechanism for concentration of urine and I will discuss the process of hemodialysis. So, if we look at for the mechanism for or to concentrate the urine, mammals has the capacity to concentrate. Now we know the basic structure, means we know there are different types of nephron in the mammals: juxtamedullary nephron, cortical nephron. Cortical nephron will have very little part in concentration of urine, but juxtamedullary nephron will have a very important role to concentrate the urine. Mammals has the capacity to concentrate urine 4 times. So here today's topic is mechanism to concentrate urine. This mechanism is also known as counter current mechanism. Counter current mechanism. So as I told that juxtamedullary nephron will have a special structure which is called vasa recta. So what is vasa recta? Vasa recta is a parallel blood vessels which will run parallel to the Henle's loop. Okay. And this vasa recta will have an important role to concentrate urine. Now, as you can see the diagram, this is Henle's loop, this one Henle's loop, and in diagram it is given the vasa recta. Now, why it is called counter current mechanism? Because the flow of vasa recta and Henle's loop is in opposite direction and they are closely associated. Means the descending limb of Henle is very closely attached to the ascending limb of vasa recta, and ascending limb of Henle is closely associated with the descending limb of vasa recta, and the flow is in the opposite direction. Now, juxtamedullary nephron, the Henle's loop of juxtamedullary nephron will go deep down into medulla. We know that kidney has 2 parts, cortex and medulla, but the Henle's loop of the juxtamedullary nephron will go deep down into medulla. And if we go deep down into medulla, we can see the osmolarity will increase. I mean, towards the cortex, it is 300 milliosmol here, then it is 600, and then it is 900 milliosmol per liter, and at the end, at the tip, at the deep down into medulla, it becomes 1200. So osmolarity will increase when we go deep down. So what is the necessity, what is the effect of increasing osmolarity? Let's discuss. Suppose there is a chamber where the solute concentration is 5 mg per liter and outside it is water, and another chamber is there where the solute concentration is much higher. For example, it is 50 mg per liter. Now, as the water is in the outside, then the rate of water which will enter into the first chamber, it will be less, but the rate of water which will come into the second chamber from outside, it will be much higher. because the concentration of solute is higher here. The same thing will happen here when the urine or the filtrate, not urine, filtrate will go deep down into the Henle's loop. Then as the concentration becomes higher, more water will come out. Here, water will come from the outside to the inside. Here, as more solute is there, more water will come out. We know the rules of osmosis. So, the effect of maintaining the higher osmolarity in the medulla is that to concentrate the urine. Okay. So, first of all, First of all, effect of increasing osmolarity. What is that? To concentrate urine. Because more and more water will come out, right? Secondly, how there will be maintaining of osmolarity, increased osmolarity by counter current mechanism. So, effect of counter current mechanism is to maintain increase osmolarity. Now, so if we need to increase the osmolarity, we need the counter current mechanism. And if we need the more and more concentrated urine, then there will be need of concentration gradient. Now, as I told you that it is a counter current mechanism. I mean, the current or the flow of the filtrate is in the opposite direction with vasa recta and Henle's loop. Now, they are closely associated. We know that descending limb of Henle, this one is descending limb of Henle. Descending limb of Henle. This one is permeable to water and impermeable to electrolytes. So, as there is high concentration of solute, high osmolarity outside, immediately there will be efflux of water. Means water will come out from the limb, right? Now, this filtrate, when it will go up by the ascending limb of Henle is permeable to electrolytes and impermeable to water. So immediately electrolytes will come out. Electrolytes here, it is nothing but NaCl. So this NaCl, I told that the vasa recta, the limb of vasa recta is closely associated. Means maybe it is present here, right? Now, if NaCl will come out, that NaCl will be readily or easily accepted, will be taken by the descending limb of vasa recta. And when the NaCl will go upward by the ascending limb of vasa recta, the NaCl will come out to the interstitium. That's how there is a very high concentration of NaCl which will be maintained here. Here means if we go down to the medulla. So concentration of NaCl will high if we go deep down into medulla. That can lead to the increased osmolarity. Not only that, from the collecting tubule will excrete, right? It will have the filtrate. It contains urea. Some amount of urea will come out from the collecting duct, and it will be there in the interstitium, and ultimately it will be absorbed by the thin segment of the ascending limb of Henle. So due to this urea, osmolarity will increase. So if we go deep down from, this is the cortex side, if we go deep down to the medulla, there will be increase of the osmolarity. And this increased osmolarity will help to extract or to extract, or we can say to absorb more and more water from the filtrate to the interstitium. Okay, that's how the counter current mechanism will work. So once again, sodium chloride will come out from the ascending limb of Henle. It will be reabsorbed by the descending limb of vasa recta, and when it will come to the ascending limb of vasa recta, it will be released into the interstitium. For sodium chloride and urea will be released from collecting duct. For that two compound, osmolarity will go, or there will be a gradient of osmolarity if we go deep down into medulla. That can help to concentrate the urine. That is nothing but counter current mechanism. Now, in the next slide, we will talk about hemodialysis. So, we will talk about hemodialysis. Hemodialysis is also called artificial kidney. It is the ultimate solution of kidney failure. Means when there is a kidney failure, kidney is not able to filtrate the blood. At that time, artificially, we can filter the blood. Means we can separate the nitrogenous waste from the blood by hemodialysis. So how it will happen? If you see this picture, you can easily understand. Now, blood from a convenient artery will be taken out here, and by the pump it will be sent to a chamber, and the chamber is filled with dialyzing fluid. So this one filled with dialyzing fluid, and you can see the coiled tube-like structure. This is made up of cellophane. This is made up of cellophane tube. So when the blood will come through the cellophane tube, this cellophane membrane allows the nitrogenous waste to pass from the tube to the dialyzing fluid, and the dialyzing fluid will have the same composition of plasma, except, I am repeating, except the nitrogenous waste will not be there. So by the process of osmosis, as they all are soluble nitrogenous waste, by the process of osmosis from the cellophane tube, it will come to the dialyzing fluid. Now here, as blood is taken out from the artery, heparin is used. Heparin is used. Why? Because, this heparin will help to prevent coagulation, and after that it will be transported or transferred to the cellophane tube, right? The blood will be transported or transferred to the cellophane tube. and it is dipped into dialyzing fluid. Now, when it will come or it will complete the circulation, when the blood will come out from the chamber, it will be free of nitrogenous waste. So when it will come out, it the bubbles, if any bubble is there is a bubble trap which can trap the bubble, and there would be addition of anti-heparin to remove the heparin from the blood. So here, when the dialyzing fluid is transferred into that chamber, dialyzing chamber, the temperature has to maintain, because directly from human body the blood will be taken out, so temperature will be maintained. So for that there will be a constant temperature bath, and there will be compression of carbon dioxide and air to maintain the blood concentration of carbon dioxide concentration of carbon dioxide and air in the blood. Now this fresh dialyzing fluid will be constantly replaced by the used dialysis fluid which will be collected here. So this is the total procedure. Most importantly, you don't need that kind of diagram, but the concept has to be very clear. So in gist, blood will be taken out from the artery. It will be pumped into a chamber where there will be dialyzing fluid, and the blood will flow through the cellophane tube. The tube, by osmosis, it can transfer the nitrogenous waste to the dialyzing fluid. In this chamber, there will be continuous supply of fresh dialyzing fluid, and it will be taken out after uses. And to maintain the temperature of the dialyzing fluid, there will be a water bath or temperature bath which can maintain the temperature. Now what will happen in the vein? When it will be transferred, blood will be transferred into the vein, this blood will not have any nitrogenous waste. So the function of kidney will be done by this unit. It will filter the nitrogenous waste from the blood. So here it is showing this is the blood which is present in the cellophane tube, and this is the dialyzing fluid. So nitrogenous waste will be transferred from the blood to the dialyzing fluid, and this is called hemodialysis. It is the ultimate solution for kidney damage, or when there is kidney is not functioning. So I hope you understood the process. Thank you for watching. For more videos, like, share and subscribe. Okay, revise well. Have a good day. Thank you. Thank you so much.